Hello, my name is Molly Gill, and my poster is about genome-wide association studies of quantitative traits in MyCode cohort. A genome-wide association study, or GWAS, is the process of finding genetic variants that are enriched in a population that has a specific trait. In order to perform a GWAS, we need a patient-linked dataset of genetic data with phenotypic markers. For this study, we performed GWASs on several traits from MyCode cohort, and we used 4 different genetic datasets that we collected from this cohort. I investigated traits such as BMI and height, as well as many lab values that can be considered markers for disease. When we plot the association of each genetic variant with the trait, we create what's called a Manhattan plot. As an example, number 3 shows the Manhattan plot for BMI. What you're looking at here is a plot of significance per genetic locus. The dotted black line represents a threshold for significance, meaning any time you see a dot above the black dotted line, there is a significant association at that locus. Inside the Manhattan plot is a QQ plot, which assesses the quality of the tests that we run. We ran similar GWASs for every trait that you see in the table at number 2 within each genetic dataset, and we compared the results we have to a public database of previously run GWASs. Number 5 is another example of a GWAS of hemoglobin A1C, which is a surrogate for diabetes. Again, we find many significant loci, and when we focus the GWAS on chromosome 6, we can find specific genes associated with the loci and assess their validity. In summary, I performed GWASs on multiple traits in the MyCode cohort, and found many genetic loci that are associated with those traits. We conclude that combining a large genetic data set with clinical data allows for associations, which can help us understand the basis for these diseases. We use a very stringent approach in our analysis, which may or may not help us to identify novel contributors. However, given our large data set, we can adjust our parameters moving forward to find additional loci. Thank you for listening.